All. Third day of the meeting, and this meeting seems to be going too quickly. We had a diversity dinner last night, which was just a celebration of our members, our diverse members, and now we have a celebration also of one of our distinguished scientists. This is the Bidder Lecture, and it's named after George Parker Bidder III, who was a marine biologist. But he also had a knack of saving journals and creating scientific societies. And way back in 1923, he saved the British Journal of Experimental Biology, and that was really the foundations for the Society for Experimental Biology. I'm really excited and delighted to introduce our speaker today. And just a reminder, our plenary lecturers recognise the outstanding contributions to their discipline. And today we have Professor Terrie Williams, who's based at the University of California, Santa Cruz. She is a wildlife ecophysiologist, does a lot of work in the field, interested in energetics, biomechanics, thermoregulation of terrestrial and aquatic mammals, and a diverse array of mammals. But they have a kind of common feature. Most of them tend to be very, very large, including elephants and narwhals and dolphins, polar bears, and Weddell seals. Today's lectures, I think, is going to be very thought-provoking. And it's going to illuminate or identify biological Achilles heels in endangered species. And by examining the functional relationships between animals and their environment, so the field really of ecophysiology, she strives to understand the ecological significance of a species and the physiological adaptations required for survival in a constantly changing world. I'd like to thank Lynn Snedden, Chair of the Animal Section, and her team, her committee, for putting forward Terrie for this lecture. So without further ado, please welcome Terrie to the stage.
Oh my gosh, I got to catch my breath. This is very, very exciting. And I want to thank the organizers, one, for an amazing meeting. I don't know if you all have gone between the different animal talks and the plant talks, but I've just learned so much. It's just brilliant. And the other thing is to be able to do this Bidder lecture. And fortunately, my second slide is already taken care of. You already know who George Parker Bidder is. And I love the fact that he's a marine biologist. I'm going to go marine today. A poet, I'm an artist, and a creative promoter of citizen science, which he did not mention, where he threw thousands of bottles with little postcards in it and asked citizens to collect these things. One was collected about 2 years ago, 108 years after the bottles were released, and it helped to define where oceans, currents were going. I find that brilliant. And as far as JEB and saving that journal basically houses my entire career. We have published extensively. My first paper was in there. We submitted one 2 days ago to the journal, so if you reviewers are out there, be kind. And everything in between, so just absolutely phenomenal. So when I was asked to do this lecture, I was told, do not focus on one study. It was to be sort of a lifetime of work. And I had to think a minute, what have I done over this life of science, 30 plus years? And we studied a lot of animals, and I came up with the one common theme beyond the animals being large. They all, in some way or another, were endangered, the potential for going extinct. And I had this thought of, my gosh, all I've done my entire career is touch extinction. And in many ways, as you'll see today, I think you all are in that same boat, and you'll find out in a second. This was also driven home about 2 years ago. We were working in Africa on African lions, and I was told that Sudan, the last northern white rhino, male white rhino, was only an hour away. And so I made everybody go to this facility, and for $40 US, you could go and touch Extinction. This animal that has since died last year, and there's something surreal about standing there realizing that you're touching essentially a dinosaur. And the thing that was most upsetting was that I knew no biology about this animal whatsoever. No metabolic rates, no cost of transports, no BMRs. You know, we know minimal about the thermal biology of these animals, and it just happened that I had a FLIR instrument that I could put on my iPhone, and I snapped this picture, and I said, well, at least I know that the top horn has a different temperature than the lower horn. But that's it, and I find that a crime. So I'm glad all you scientists are here, because this is what we're up against. And all of you, especially if you're dealing with animals that I know about, but I'm sure this is the same for inverts as well as the verts, and for plants, that over history, when you start recording the extinction rates for animals, that dotted line, let's see if I can do this. This dotted line is what extinction is supposed to be doing naturally, and look at where we are these days. So there's a very good chance you're going to go out there, be studying an animal, and by the time you finish your career, that animal could be gone or on the verge of gone unless you've done something right. So just keep that in mind. So who do we study and who do we save, and is there some way that we can predict, you know, who's going to make it and who's not going to make it just based on animal design? And there are beautiful designs throughout the animal kingdom, and of these, are there some that are so poorly designed that sooner or later they're going to blink out, some that are going to blink out really quickly, others that, you know, may have such a perfect design that they can withstand all the things that are going on these days. And that's sort of been the goal of our science more recently than ever, to try and figure out what are the biological attributes that are going to allow for various animals of different design to survive. So today, in honor of Bitter, what I'm going to do is focus on the marine side and look at the challenges to animal design that have occurred in one of the more remarkable types of animal transitions, and that's the 60 million year transition of a quadrupedal animal that used to walk around on land and turn that into some of the biggest vertebrates ever known that now swim in the oceans. And if we look at sort of a simple phylogenetic tree, and that's what I've shown here for mostly carnivorous animals and some of the hooved animals, we find that there are essentially seven different lineages that ended up in going from that terrestrial to a marine adapted animal. So we have the sea otters on top, the pinnipeds coming in after that. We've got polar bears, which are marine mammals, and I'll prove that today, and the odontocetes, and then mysticete whales, and finally the dugongs and sirenians. So 7 different times terrestrial mammals have turned to aquatic habitats. And what I'm going to do today is sort of take the extremes. We're going to look at a couple of the more recent animals, so the sea otter and the polar bear, that still have a bit of terrestrial design to them, but they live in aquatic environments, came around 10 million years ago to about 1 million years ago, a little bit less. And then some of the more ancient lineages, when we start talking about the great whales and dugongs and manatees, there you're talking 60 to 50 million years of evolution behind the designs of those animals, highly adapted 100% of the time for aquatic living. And here are some of the major challenges. You've got metabolic challenges, locomotor, thermal, hypoxic. These are the things that you had to change in a terrestrial animal to turn it into an aquatic mammal. And we're going to do 3 different animals, just for the sake of time. We're going to take a look at 2 recent arrivals, our polar bears and our sea otters, look at feeding the metabolic machine for living in aquatic environments, warming that metabolic machine, and then finally looking at some highly adapted mammals for living in the marine environment, and that's the odontocetes that have learned how to submerge that metabolic machine. And if we do just the simple biology, if we do nothing more than just compare the basic metabolic rates of marine mammals versus terrestrial mammals, this is the kind of graph that we've developed over about 30-some years. And the bottom line is marine living is energetically costly to animals. The resting metabolic rate of our marine mammals, all shown along that dashed line, from sea otter all the way up to the great whales, they have a metabolic rate that's about, on average, 2.5 times that of terrestrial carnivores predicted by the Kleiber equation, which is shown by the dashed line. So just keep it in the back of your head. It's tough energetically to be a marine mammal. Now, the thing I find so fascinating, this is why I love science, this paper came out just a week ago, maybe 10 days ago, and in it, it was talking about humans, and it said, what is the sort of maximum human energy expenditure for long periods of time? And in the end, it turned out that the energy intake of humans, on average, for long, sustained, maximal exercise is about 2 times your BMR. And I find it fascinating that the marine animals get to that 2.5 level times a basal metabolic rate, and that's about it. This is a maximum. Except for the marine mammals, they're only resting at that point. So they've got to go over and above that by about 3 times to start to look at what humans are doing. The thing I also found fascinating, that the limit for humans was simply that you can't process enough calories to get above that 2.5 times BMR. All right, that's the max that you can bring out and sustain for a period of time. And interestingly enough, for the marine mammals, that was the limit. That was a defining factor for our marine mammals. We take our phylogenetic tree that's shown here. We've color-coded it now for the intestinal length divided by the body length. And so the cool colors, the blues and such, are lineages that never developed a marine form. And so that's the felids and the canids. Some of the horses never became a marine mammal. All of those lineages that are in the green and the yellows and the oranges and the reds have enormously long small intestines. They are processing a lot of calories in order to be a marine mammal. So that design to bring in lots of food and process it was how we got to these 7 different lineages of marine mammals. So let's see how they feed that metabolic machine. And we're going to look at one of the biggest feeders that are out there, and that's the polar bear. We started looking at the energetic cost, not just of resting, but of swimming. We built a swimming flume. So this is a bear at the Oregon Zoo. We've baited it into the flume with fish. And then there's a current, and he's just going to swim against that current. And there's a metabolic chamber on top of that, and we're measuring the metabolic rate of that polar bear swimming. And what we found is that using that kind of technology for polar bears and manatees and dolphins and sea otters and mink and such, it really depends on how well your body is designed to swim either on top of the water, below the water, and what kind of paddle you're using. So for the true dog paddling kinds of animals, and the sea otter falls into that, it's a rower technically, but for the mink, the otters, and for the polar bear, their cost per stroke is infinitely higher than our undulators that are swimming underwater, including all of the dolphins and the manatee and the human. I don't care about Michael Phelps. We're just not that good at swimming. Here's a polar bear that we trained to walk on a treadmill. The trick, I'm showing you here, this is training it. The trick is baby food. That's what we're baiting the bear with. And he learns to walk on that. That's a metabolic chamber that's around the animal. We eventually close the entire thing off, get it walking at higher speeds, and then we run everything to Seville systems, O2 analyzer, and figure out the cost of walking. And without going into a lot of details, it turns out that that walking behavior is the same cost as you would for any quadruped of that size. So nothing special there except when the bear tries to run. Exceedingly high cost for running, and we don't see very, very fast speeds for polar bears for long periods of time on the sea ice. They're not, their gait just isn't built for that, and certainly it costs them. So when we were running those experiments, we would calibrate our smart collars. And these are collars that have a GPS attached to accelerometers, magnetometers, cameras, and other tags that we were using for some of the other marine mammals. But the idea is that we would know the oxygen consumption that goes with the G-forces from the accelerometer, and then in using our collars in the wild, we could get very discrete energetic costs measured for the animals by simply having a collar on them. And that's what my graduate student, Anthony Pagano, did. He took these collars, we got them calibrated both for behavior and for the energetics, and deployed them on polar bears up in the Bering Sea. So the brown part of this graph is the land. This is the top of Alaska. And then the white is the sea ice that the animals are walking across, and all of those tracks are where the animals are going. And the thing that was probably the most fascinating in this is that these animals did a lot of walking and didn't get very far. And the reason for that is that you have to remember, and especially now, that ice is moving constantly underneath the bear's feet. And it's actually trying to stay in one place. Oh, hang on, I'm a little ahead of myself. So first thing is that, not surprisingly, the field metabolic rates of these animals increase either the faster they're moving or the longer that they move. So walking costs these bears. They're just not getting very far. And the reason they're not getting very far is the one thing the bears are trying to do is stay in this ring seal habitat, which tends to be a fairly coastal place. And they're going to go out to try and find these seals. This is their main prey item. But when the ice is moving, and especially now that things are getting warm, the ice is moving a lot, they're having to walk an extraordinarily long periods of time in order to just stay in one place to feed on the major prey items. And here's a video camera showing the accelerometer traces on the bottom. Those big things are where the bear is just ripping the heck out of this ring seal that it's caught. And the thing that we noticed, and it's just so apparent, the only thing the bear really cares about is fat. It wants to eat the blubber. The rest of the animal, the viscera and the muscle, is secondary. The thing it wants to do is essentially skin out that animal and eat that high caloric density fat. And therein lies the polar bear's Achilles heel. In order for this animal to live in the environment that it lives in above the Arctic Circle, it has to have fat. And it does that by eating seal blubber, and it does it by eating bowhead whale blubber from carcasses that are on a beach that local natives may have killed. And that's what sustains these bears. So when Anthony went out and was collaring these animals, we found that about half the bears lost their body mass over just a short period of time. We're talking weeks to a month here. And already we've got 4 bears that lost body mass, 4 bears that increased their body mass, 1 that didn't change body mass, and the ones that looked like this bear walking around looking really poor were the ones that chose, or I shouldn't say chose, but only found food that was muscle food or just scavenging carcasses. So all of those animals, let's see if I can do this. The animals that could find an adult or subadult ring seal, so in the black here, those are the animals that are eating the prime food for a polar bear, eating all of that fat, and as a result, they were surviving. The ones that couldn't find it or were scavenging on bowhead whales, as good as the fat may be there, oftentimes there wasn't enough to sustain the animal. And here's the problem. What could go wrong? What has happened over the years is the difference between what you see in the white zone there for 2018 for the edge of the sea ice that's coming to the top of Alaska where we're studying our animals. And this is what it used to look like in yellow, and this is what the bears are encountering now. So we have a situation in which bears are having to make a decision. They have to decide to stay with the sea ice, go out to sea on the ice, which will move them out of those ring seal areas for eating, or learn how to eat on land. And the options on land are not great for them. And Anthony actually is first author on a paper that we have in review in Ecology right now, and I'm not going to steal his thunder, so you're going to have to wait and see what the bears decide. So let's talk about warming the metabolic machine. If there ever was an animal that should be extinct, it should be the sea otter. And it's because you've got the smallest marine mammal with the highest surface to volume ratio for losing heat, wearing a fur coat in cold water in Alaska, and it has no blubber layer. This is an animal that has to eat an enormous amount of food just to sustain itself, to rest in water. And I truly don't understand why these things haven't gone extinct, because they are truly, truly on the edge. This is what a marine mammal should look like. This is like the ring seal, and in fact, it's a harbor seal that's been cut in half. And that white area all around the edge is the blubber layer of that animal. That's great insulation. It's the way that pinnipeds and cetaceans stay warm in the water. It is not what sea otters do. Sea otters don't have this air or this blubber layer at all. But they do have the densest fur of any mammal that's been measured to date, 26,000 to 165,000 hairs per square centimeter. It is amazing fur. It is fur that they've been almost wiped out for because of the density of it and the fineness of it. And there's a picture of the cross-sectional view and then the microscopic view. It is remarkable. And also, the thing that we've discovered, we did a bunch of electron micrographs, and we found that it's the cuticles on the hair that have really changed in terms of aquatic mammals versus terrestrial mammals. Very smooth hair shafts for something like a coyote. You can see in the fur seal the kinds of cuticles that have developed there. We see it in the river otter. We don't see it for the California sea lion that doesn't use their fur for insulation. And the sea otters are absolutely king when it comes to trying to keep warm by using fur in water. And what the sea otter does, I don't know if any of you have ever watched one of these animals, but it's in constant motion, and it's rubbing its fur, and it's breathing air into its fur and the rest of it. And it spends a large proportion of its day just grooming that fur, trying to get an air layer in those hairs. And it's actually felting the individual hairs to create almost like a wetsuit situation for the animal. It's actually an air layer that keeps the animals warm. But you can see from the metabolic costs here, grooming is the next highest cost for these animals after just resting and feeding, more so than swimming. So this is a costly way to try and stay warm. And in fact, this is work that was done by Laura Yates, who implanted an abdominal temperature-sensitive transmitters in sea otters out in the wild. Otters, 24 hours a day, are on a roller coaster of changes in their core body temperature, and that's what this red line is showing you. They go through a cycle of feeding and grooming and resting and foraging and unknown, but it's basically the animal grooming, then resting, then foraging, grooming, and resting. And they have to do this 24 hours a day. And the body temperature is going up and down depending on what behavior they've gotten to. And so if they've fed and they get here, they groom the body, and as they're resting, that heat increment of feeding, SDA effect, happens here. The animal's still resting, body temperature comes down. And once it reaches about this 37.8, the animal wakes up and it's got to start the cycle all over again. So it has to have food constantly available to keep these fires going and to keep itself warm. And that's the otter's Achilles heel. They have an incredible thermal lability going on, and you can't break this up. So what can go wrong? Things went really wrong on March 24th, 1989, when the Exxon Valdez spill happened, dumped 11 million gallons of oil into Prince William Sound, where there are thousands and thousands of sea otters in that oil spill zone. And fortunately or unfortunately, I was called by the US Department of Interior because we had studied metabolic rates in sea otters, and they said, we want you to come up here and fix the situation. It was a life changer because suddenly they wanted us to go out, capture these animals, bring them in, clean them up, and send them back out again. And it was the most insane thing I had ever done. And being a scientist, I decided we were not just going to do that. We were going to record everything, including what had happened to the internal organs of these animals, to figure out what went wrong when an otter got oiled. And it actually led to the first of 3 arrest warrants for me during the spill. We'll have to drink a beer over those. But to get, to get an idea of how oiled animals get, otters are always brown, but I came up there. We also were dealing with harbor seals and harbor seal pups. This is a harbor seal that came in. I thought harbor seals were black or brown in Alaska. This is before cleaning, and this is after cleaning. So you get a sense of the amount of oil that gets on these animals affects their fur. Harbor seals don't have an insulation problem because they've got all the fat. So cleaning them off was easy. Otters, it was a whole different story because they lack that fat layer. This is an infrared thermograph of when insulation works for a sea otter. All those red areas are the front paws and the back paws. The fur keeps the animal warm normally. If the animal is oiled, it destroys that insulation, and it destroyed it very quickly. And what happened was the animals lost core body temperature very, very fast. This is the biggest problem that we were dealing with in terms of the animals that were impacted by the spill. They simply went hypothermic. So all of these people that were thinking of a toxic hit, and there were issues related to that, the biggest thing that we were fighting were the cardiovascular changes that happen when an animal goes hypothermic. So the liver, the kidneys, the gastrointestinal tracts were shut down, led to congestion. There's a picture of the lining of the stomach. We thought that was oil. It wasn't. It was blood. It was focal hemorrhaging that was happening in the gastrointestinal system because of the cardiovascular shutdown with the hypothermia. These animals simply got cold, and in fact, 50 to 80% of the animals were dead as a result of that. In the end, it's sort of funny, it comes back to the paper I just referred to early on. There are not enough hours in a day for an oiled otter to groom, forage, and process food to compensate for that heat loss. And that was the reason we were losing so many of these animals. So a solution is to just submerge the metabolic machine. And this is the most recent work that we've been doing. The animals that have escaped a lot of the problems that I just talked about for the sea otter and for the polar bear are the ones that are the most highly adapted, and the way that they've adapted themselves is to submerge that metabolic machinery. And that takes a lot of physiology to get to the animals to be able to do this. We've been, as we've been studying these animals, we've been looking at the deepest divers. The way that marine mammals get sort of are able to submerge that machine is they have to develop a scuba tank. They have to be able to have an internal oxygen in their blood, in their muscles primarily. They collapse down the lungs. And so you can see for our deepest divers, narwhals and sperm whales, really high levels of oxygen in their tissues. And same thing when we get to Weddell seals, hooded seals, and our elephant seals. These animals have enormous oxygen stores that enable them to hold their breath for long periods of time. In doing so, they actually avoid oil spills and that kind of thing, so we don't have an issue of surface pollutants for them. And they can follow their food resources better than a walking polar bear or even a swimming polar bear. Now, the other side of it, though, is that even if you have a scuba tank, you want to be really efficient in what you're doing as far as the costs of locomotion and the costs of your metabolism, because you'll use that scuba tank very quickly. And this was a project that we were doing looking at Weddell seals in the Antarctic, and my colleagues and I had developed this camera system where a camera was put onto the Weddell seal's head, and we were supposed to be looking at behaviors, so foraging, what kind of fish they were taking. And it was just one of those moments where I said, let's take the camera and turn it around. I just want to know what they're doing swimming-wise. This was before the accelerometers had been developed. And my colleagues were so mad at me for wanting to turn and waste one of our deployments on this backwards kind of deployment where we were going to look at the butt of the animal. And what we saw was actually nothing, because nothing was happening on the back end of the animals. And this was, it took a moment to process what was happening. What we realized in these films were for minutes and minutes and minutes during the course of the dive, the flippers never took a stroke. We started to realize that marine mammals cheat, that they're able to take that scuba tank and extend the life of it by simply not swimming, not taking a stroke, and turning off that metabolic machinery any way that they can. So the red actually shows you the gliding periods for these animals. There's not a stroke taken during that entire period. They start off stroking, get themselves going, gravity takes over as the lungs collapse, they come down to the bottom of the dive, and then they stroke or stroke and glide to the top. And the remarkable thing is that Weddell seals did it, and so did elephant seals, bottlenose dolphins, and blue whales. And there are variations on a theme for this. Right whales are in the opposite direction because they're so buoyant. But the bottom line is that these animals use different gaits, and they try to glide as much as they can during the course of their diving. If I can do this. So now we come to narwhals. Narwhals have been this iconic animal that I didn't even believe existed. And I wanted to study them because it was a deep diving cetacean. We'd been working with bottlenose dolphins, we had worked with these deep diving Weddell seals, and I wanted to get to a point where we could do something with any cetacean whatsoever, and an opportunity came up to look at these animals. Now, a narwhal is one of the deepest diving of the marine mammals. It can dive to about 1,500 meters, and it's up there with the beaked whales. Those are pelagic whales that are out to sea. We just can't get our hands on these other species that are deep divers, and sperm whales, too big for me. Even I have my limits. But the narwhal, we can literally get our hands on. And the reason we can do that is once a year, about the month of late July and into August, and I'll be heading out to Greenland in August, we can work with local hunters. These animals have come into a fjord system, they're in a coastal region, and we can put our instrumentation on them. So what you're seeing in that picture is one of our heart rate recorders. It actually is a depth meter, an accelerometer that counts strokes, and a heart rate recorder. It's in a housing that will float when it comes off. All we're doing is suction cupping these things onto the backs of the animals. And then we have a satellite tag and a radio tag that will enable us to pick the instrument up. There's a picture of the animal being deployed. There's a graph of a typical EKG. And then there's an area in Greenland on the east coast that we're working. So a dense area of fjords. Unfortunately, an area that's becoming more and more ice-free, and that whole east coast is now up for potential oil development. So you can imagine there's intense interest by both Greenland and Denmark, who's responsible for the population of narwhals and their management, wanting to know, what's going to happen to these animals and to have some basic biology done. This is a situation when we're working with the narwhals. You notice I'm not standing in front of that long tusk. The two guys are standing there. It's like, duh. It's Mads Peter is the person who runs the program, so he provides the logistical support. Susanna Blackwell is an acoustician that is part of our team. And the situation is one in which the narwhals are hunting in this area for narwhals. They take the muktuk from them and sell it. But some of the animals are given for science, and we release the animals from the nets after we've put our instrumentation on, and then the animals are free to do whatever narwhals are going to do. For us, my instrumentation, because it's suction cupped on, will stay on for about 4 days. And then we have to use the tracking technology in order to find our tags in amongst all the icebergs. And generally, we've been really lucky. We've only lost one of these things, despite the fact that it can get into some really remote areas that these narwhals are getting into. So that's the heart rate of a narwhal. And it's the heart rate of the animal as it's resting on the water surface. We're walking around it, so it's talking back to us, but you get a sense that it's not a really slow heart rate. Even though it's submerged there, it's in shallow water, heart rate is about 60 beats per minute. This is one of these aha moments, so this is mostly for grad students more than anything else. So this is the first record we got back from one of the narwhals, and it's showing you heart rate in relation to just time. So these are the first 5 dives for a narwhal. It had been in a net. We put our instruments on. We sent it out. And I almost threw this data away because I said, well, this isn't normal. This is an animal, 60 beats per minute that you just heard. It's mucking around here. It's trying to roll and get away from us, comes up to the water surface, comes down, does some crazy dives. And then eventually, after about 2 hours, the heart rate gets back into a normal pattern, and then we've got what we consider normal diving behavior and foraging behavior by the animals. And you want to throw it away and say, well, this is abnormal stuff, and we're going to stick with all the normal and tell you about what a narwhal's heart rate is normally doing. And then I realized that no, this is actually the important stuff. This is what a narwhal does when it's trying to escape. And what it did was something I've never seen before, and that was a couple of dives where I've never seen a heart rate this low for an animal this large, 3 and 4 beats per minute. It was remarkable, and it did that as a part of the escape from all of us. So an extreme bradycardia. So this is, and I'm going to have to ask some help for here, so hold on, don't do it yet. So this is what 3 to 4 beats per minute sounds like. So if somebody in the back can hit that little icon, maybe not. It's a little speaker. Nope, go back. No, keep going. Go back to the original slide. One more. Right there in the lower right hand. Yes, hit that with your, perfect. That's the first beat. Now I'm going to talk. No, don't touch it. You're listening to the interbeat interval for that animal at 3 and 4 beats per minute. And so, assuming it's still going, there's a second beat. Your heart has beat 20 times in that interval, and this animal's trying to move blood around its body at 3 and 4 beats per minute as it's escaping from us. Absolutely remarkable. Whoops. There. So what we realized is that these animals have a pretty paradoxical escape response in these deep divers. They have a dive response. They have an exercise response happening at the same time, because remember, they're trying to get away from us. They have a fear response. All of these things are superimposed on one another, and look at what it's doing to the heart. The dive response is saying go into a bradycardia. The exercise response is saying increase your heart rate because you're exercising. The fear response is saying go into an extreme bradycardia. And on top of that, the cognitive control over heart rate and an adrenergic input. What's a heart to do with all of this input? We actually found a lot of cardiac arrhythmias that were occurring in these animals and in extreme diving animals when submerged, trying to swim really, really quickly. And therein is the narwhal's Achilles heel. It has so much going on in trying to escape any kind of aversive stimuli. The fascinating thing is that I was once stupid enough to try an Ironman kind of competition. I'm like way back here in a pink cap. And this kind of situation to me is exactly what these narwhals are up against. You are standing there, a cannon goes off, your heart is going as high as it can, you're terrified because all of these people are going to pile on top of you, you're going to hit the water, your face is going to get cold, you're going to go into a dive response. So you got fear, dive, and exercise all happening at the same time. And in fact, in these cold water mass start swims, humans have died. This is the one part of a triathlon that is actually the most dangerous in terms of race day deaths. And I think, or I shouldn't say just me, but we think that this paradoxical combination of diving fear and exercise is something that is really difficult to do to your heart. And so they've changed the starts of a lot of these Ironman competitions by making it a slow, controlled start to get rid of that fear factor. And it's helped to really, if nothing else, reduce the stress. But the idea was try to reduce these cardiac conflicts that can happen. And they still do happen with people that are afraid of swimming and do this kind of thing. So what could go wrong? Well, a seismic ship came into Scoresby Sound 2 years in a row for our experiments. And we suddenly had the threat of a new kind of stimuli on these deep diving animals. This is a picture. The red heart is one of our tagged narwhals. The green is a seismic ship that's come into the fjord, and in a second, that ship is going to run right over where our narwhal wants to dive, and this is what it hears. So that is a single seismic pulse. Those kinds of pulses from oil exploration ships go on anywhere between 15 to 40 pulses a minute. This is what these animals are being exposed to. This is what that recorder that we deployed on the animals are showing us. Oops. We get breathing rates after the animals come to the surface. The arrows are showing you the QRS peaks from the EKG of the animal, and then we have stroking rates. In my entire life, I have never seen such extreme physiology. This animal's heart rate, when it gets to the surface after an event like this, there is no interbeat interval. I don't think it can get any higher, and they certainly can't stroke any higher. And in a second, I'll show you they can't breathe any faster. And so here's the effect of having that kind of noise in the environment with these animals. The top picture is showing you exercise levels, so stroking, that's color-coded. The bottom is showing you the depth of the animal. And what's happening is, under normal circumstances, we have an animal, it's got a pretty moderate stroke frequency. Here's that glide I was telling you about, so that's in white. Low stroking, a little bit higher to get the animal going up on the dive, and that's normal. When there is a seismic explosion, the animal's swimming really, really fast, it swims down during the course of the dive, it's still stroking here. One little period of gliding, stroking, stroking, stroking fast on the bottom, really powering up, and then doing the thing over and over again to escape that sound. The heart rate is following suit in these activities. So the top is showing you normal situation for the heart rate. This is that extended dive during exposure. The animal's heart, though, is plummeting. It's getting down, reducing that heart rate into that fear response. Here's that cardiac freeze of 3 to 4 beats per minute, and then it is screaming on the way up. The thing to notice here is exercising really hard, cardiac freeze there, and I'm going to show you this in a different way, just so that you can see. So here's normal response for the animals. Glide a whole lot here, this is pre-exposure, and then have some stroke frequencies to let the animal get through the dive. Here's what heart rate does, lots of low heart rates because it's not swimming that hard, and then a series of strokes as it's, or a series of increased heart rates as the animal is stroking more. Here's what happens during exposure, and this is what I find the most frightening. So here in red, superimposed on our situation in gray, the animal gets away from any gliding at all, and it is now exercising as hard as it can, but at the same time, it's going into a cardiac freeze. So high exercise for this animal underwater while its heart is providing the least amount of blood during the course of that dive. This is a recipe for a problem. Its heart rate during that recovery, as I was said, an animal like this having an 80 plus to 90 plus beat per minute heart rate, I just haven't seen anything quite like that. And as I said, there's almost no interbeat interval in terms of any blank period between each beat. I don't think it can go any higher. Diving frequency, this is just, or sorry, not diving, respiratory frequency during the recovery. Essentially, if the animal dives for a short period of time, it's okay, but their response is to increase the duration of the dive to get away from these sounds, and I think we've hit a maximum respiratory rate for this species during these exposures. The metabolic demands are huge. I'll show you a normal dive right here. The animal uses about half of its O2 store, so this is like that SCUBA tank, and so generally it'll have a cost right here, uses about half of its SCUBA tank. This is what happens when there's a seismic pulse. It's so far beyond its ADL, it's emptied the SCUBA tank, so they have to be going anaerobic, and the costs are over, it's just enormous, 3 to 6 times what the animal normally expends for a similarly long dive. This is what's going on. We've got all of this activity happening in terms of fear and exercise and diving. And I believe that we're getting into a situation of brain hypoxia challenge. And we actually, for now, are doing an entirely new sort of set of experiments looking at brain function in marine mammals. And it's because of this effect of noise on the animals. It's not just the physiological effects. I believe that there are also physical effects associated with this kind of noise. And you have to remember, this is an animal, like a human, you got 82 billion neurons that have to be firing flawlessly when you're underwater. You can't have a mistake and you can't have a stroke underwater because you will be dead. The number of times that animals have come up stranded as a result of noise in the oceans have involved mostly these deep diving whales. You don't hear about pinnipeds coming up that often, and certainly the smaller cetaceans. Normally in a stranding event, it tends to be a situation in which some disease or thermal component is in there. When you get these deep pelagic whales coming in large numbers, we suspect a noise event has happened. A lot of these military sonars have been implicated. And the thing that's so curious about it is that if you hear about these people trying to push these animals back out to sea and they go, we don't understand, the animals swam right back, they must be stupid or something. They're confused, they're disoriented, and I think that may be a clue about this central nervous system hit that they may be undergoing. Are all brains the same? I can tell you that in the design of going from terrestrial mammals to marine mammals, a lot of things have changed, and not the least of which is how the brain is neuroprotected, either by the cardiovascular system and capillary density. Certainly blood flow is different, and it's going to be, you'll hear about this in the next couple of years, I think. So, what did I tell you today? I gave you 3 different animal designs. We went through 2 groups that were, pretty recent to the marine environment, and a group that is absolutely, adapted for the marine environment. They all had an Achilles heel. For the bear, it was how they process large amounts of fat. For the sea otter, it was its fur. And for the narwhals, it was this sort of paradoxical response it has to fear, diving, and exercise when submerged. The physiological response in the wild is that those bears have to have the right amounts of fat in prey items in order to survive. The sea otter has this huge liability going on in terms of its core body temperature, and again, it has to have food available. And the narwhal, very extreme responses to aversive stimuli. The threat, they're the same threats that you've heard for so many animals. The threat is loss of ice for the polar bear, pollution for the sea otter, and oceanic noise right now for a lot of these deep diving animals. And the potential for survival, that I didn't mean for this to happen this way, but it's really interesting that for at least these ocean animals, it's fossil fuel. Whether we are, creating climate change, changing the ice situation, whether we are spilling oil in the oceans, or whether we're just looking for oil in the oceans, this is a bad place to go and try and find our next energy source. So in terms of helping these animals, maybe that's a place to start on top of the science we're doing. Why should you care? Why should any of us care about this? We got into this because of the love of nature, and I believe that most humans do love nature, whether it's the plants you study or the animals that we study here. But here's the thing. We hear these words all the time, and as scientists, we're not immune. We know what it means when you talk about habitat destruction and climate change and disease, resource limitations, acidification, melting ice, you name it. The difference is that we're on the ground, we're studying these animals, we are witness to the effects of what's going on here. And I don't think most people, in the world have an idea of what habitat destruction really means. What does it mean to the individual animal and populations of animals? And I think that's our job. We are there, we're seeing it. We are the ones that are touching this extinction, and I don't think you want to be in this situation. I don't want to be in this situation again, where you're holding on, getting your picture taken with an animal that is essentially gone, and it's happening too often. So if I can say anything to any of the folks here, and especially the young folks, your science is more critical than ever right now, because I think science is the truth. Science is going to get us to solve these problems, and we got to get people to understand what is happening on the ground. So I'm going to show you why you don't want an otter to get oiled. This is some archival footage, and I want to show you what happened during the Exxon Valdez oil spill, and they brought in these animals. This was a life changer in my work. when all I was supposed to be was a scientist, and suddenly I'm in charge of taking care of oiled animals. We knew how to clean them, and we thought that that was going to solve the problem, but it was the science that led us to how to really know what the oil was doing. So hours and hours of scrubbing. Dawn detergent works the best, I will tell you, give a plug for them. But these were people that were off the street that we were just leading to try and solve the problem for this otter. And the reason these otters aren't chewing the heck out of us is because they were hypothermic. And so they'd get woken up when we would put warm water on them. But they really were in a bad way. And a lot of them were starving after just a few days of not being able to forage. So you have to wash the animals, you gotta rinse the animals because you're trying to get all of that detergent out of a fur that has, you know, this amazing density to it, and it took a long time. This old guy was a bugger. I got a scar from him. So you would have to look at this fur. The amazing thing about sea otter fur is that it starts to loft up, but trying to check 300,000 hairs per square inch is not easy. And eventually, you got to the point of being able to dry the animals off. Took another hour, so you've got a total of over 5 hours for every single otter. And in the end, we were able to release about, see, the number's going to come up, 259 of these animals back into the wild. And these are the people that helped me out with all of these things. These are some of my funders, and I thank you for your attention, and you can go eat lunch now. All right, thank you very much. All right.